So I push the button. Hopefully I see the red button up here, red icon. So this is recording. So after this overview of the muscles, let's take a look at the visual aspect of this. And I see another question here. What lab report are you working from? Did you find it? This is in the lab reports and supporting information. Has anybody found the lab report, or is this something I forgot to attach? Which is not impossible, but I'm waiting for you to tell me.
Is it the muscle and nervous system and integumentary system
That's correct. That's
lab report? Okay, thank you.
Yes. So apparently it's there. That's great. Let me do this screen share, and we're going to go to the lab shell. And before we get to this, we're going to take a look at this. I'm going to try to scroll this up, and let me see if I can make this a little smaller. So for overview, here's what skeletal muscles look like under the microscope. Where are the cells? We are seeing these logs. So where are these logs? The middle of the skeletal muscle cells. Here's one log. They're kind of diagonal. Here is the second muscle cell, or part of it. The third one here is a lot skinnier. Here's the fourth one. Here's the fifth one. Again, cells go from left to right on this field. And you can see more cells here. Again, diagonally right on top of each other. You see a little gap between muscle cells here. So when you look at this tissue page, the location of this tissue is in skeletal muscles, and function is voluntary movement of bones and skin. In the muscle lab, we will talk about muscles of facial expression, and you'll see that we have a whole bunch of skeletal muscles that are moving the skin around our face, whether it's smiling, closing our eyes, raising the upper lip. We have muscles for that, and we have to move skin to move those skin parts to create a facial expression. So what I'm going to attempt here is to zoom in on the image, and I hope I won't lose anybody in this process. So here's a zoomed in version. Under a microscope, this would be super crisp. So here is the striation, especially on the skinny muscle cell here. You can see these lines perpendicular to the muscle cell's axis. These are very, very thin lines, but you can see hundreds of them. You can see it on this muscle cell too, one above it here too, maybe to the left. Those are the lines representing striations. And again, these are repeated structures inside a muscle cell. It all has to do with contraction. And we'll come back to this in the first muscle lab, just what is forming the striations. So this is the reason why the skeletal muscles are called the striated muscle. It's also a good view. Take a look at this muscle cell here. And you can see several nuclei. Here's 1, 2, 3, 4, 5, 6, 7. So this is why a muscle cell is multinucleated. It has several nuclei. And this is a unique property of skeletal muscle. I'm going to try to go back. Any questions about skeletal muscle? Our next issue, let's go to cardiac muscle. And if I can scroll this up and maybe zoom out to show all the lines here. So when you look at this muscle in this view, it's just a purple mass of cells. I'm going to zoom in because if we would project this in the lab, a much bigger image would be the result, and details would come out a lot more crisp. Location is the heart only. Function is involuntary pumping of the heart. You can put down involuntary contraction of ventricles, atria, or heart chambers. Let's take a look at in a zoomed in view, and we are looking for intercalated discs. They are visible. So let me see if I can zoom in here. The muscle cells have three-dimensional branching, and so the individual cells no longer show up very crisp like the skeletal muscle. But here's one muscle cell. Look at this rectangle. Here's the second one, but the rest of it is obscured. Here's another muscle cell. Here's the beginning, and the muscle cell goes here, branches out this way. At the beginning, at the end of muscle cells, we see these dark stick-shaped lines. These are the intercalated discs. From a tissue structure point of view, these are desmosomes. They are gluing neighboring muscle cells together. And if you think about it, in our heart, there are no bones. There's no tendon or bone that the muscle could grab onto or hold onto. But the muscle cells are holding onto each other. And you see quite a few of these intercalated discs. Here's another one, a little skinnier here, but here too. In the distance, there's another intercalated disc. So all these lines, dark brown or dark purple sticks, are intercalated discs. And that's the best we can say about cardiac muscle, again, because of the overlapping and underlapping of the cells. There's not a great crispness to this tissue. This is a great illustration. Cardiac muscle is striated. We cannot see this, even in the zoomed in state.
Are intercalated discs the same as desmosomes? Like if you had an arrow on the test pointing to this line, would both answers be correct, or?
I would take both, but the proper name of this is intercalated disc. These discs were named before desmosomes were discovered, before anybody knew what they were, just like the striation. When histologists looked at this tissue and saw these sticks, they felt compelled to give it a name. And it was later found out that these are desmosomes. So the proper name would be intercalated discs. It is a type of desmosome, but we have desmosomes on epithelial cells as well. So let me zoom back in here. Any questions about cardiac muscle? Our last tissue here is smooth muscle. So this is where the spindle shape will be more visible. I'm going to zoom in a minute. Really important words here: location, walls of hollow organs. Smooth muscle is inside the walls of many organs, inside the walls of blood vessels. What could be these hollow organs? Think about organs of the digestive system, esophagus, small intestine, large intestine. Think about some parts of our respiratory system. Bronchioles have smooth muscle content. What about reproductive system? Maybe the most familiar, something that we looked at on those models in lab 1, uterus is especially rich in smooth muscle. Vas deferens or ductus deferens in the male reproductive system also has smooth muscle, but they're all in the walls of these organs. So this is the best way to express location of smooth muscles. In later on today, in the skin, we will observe a smooth muscle structure. So that's just as good answer as these two here. This tiny smooth muscle in our skin called arrector pili muscle. We'll see that in a little bit. So that's a good answer here for location of smooth muscle as well. Function is, again, please pick one you might want to highlight or circle because one out of many functions is good enough. Involuntary contraction or involuntary movement of food, blood, urine, or bile. So this is referring to those hollow organs, movement of food, stomach, small, large intestine, movement of blood. Our blood vessels can move blood along using smooth muscle contractions. Movement of urine is the result of smooth muscles contracting in our urinary bladder. And movement of the bile is consequence of smooth muscle contracting the gallbladder. So these are all organs that we have met on our mini torso or on a pelvic model. And so this is just a small segment of what kind of materials are moved by smooth muscle. Again, we mentioned this earlier, this is all involuntary. We have no conscious control over smooth muscle movement. I am going to zoom in just for a few seconds. This is a non-striated tissue, so we're not looking for striations, but the really long spindle shape of smooth muscle cells are maybe visible here. You can see this single nucleus, and here's one muscle cell. The borders are hard to see, but this muscle cell goes all the way here, really sharp spindle shape. We have several dozens of these smooth muscle cells piled on top of each other to form a bundle of these smooth muscles. So this is the appearance of smooth muscle under our microscopes. Any questions about this last muscle tissue?
What does spindle shape mean? I thought a spindle shape was like eat an apple. These look like long and noodle-like.
Yes, exactly. Or some people would say a stretched football shape, the tapered ends and pulled out to long distances. You can think about a cigar. That's another way to phrase this, with the tapered ends. So long, skinny structures. The spindle fibers are like that. We mentioned those in cell division. We didn't have a really good look at them, but those are, again, long, thin structures. The skeletal muscle cell, for example, has the dimensions of 5 to 8 inches in length for the width of a human hair. So that is called a spindle shape. Smooth muscle cells, a little shorter, yet under the microscope, they're still belie this long, thin shape. So that's what the spindle shape refers to. Any other questions about muscles, muscle tissue, smooth cardiac, skeletal muscle? give me a chance to check any chats that may have come in. I don't see anything new. So I'm going to come back to this shared lab shell. So there's one more tissue category left, and that is nervous tissue. When you look at your lab report, the nervous tissue has a few more attributes here. Let's say a few words about what qualifies nervous tissue as its own category. So nervous tissue is made up of two groups of cells that like night and day, but there are some really giant dominating cells in nervous tissue, and those are called the nerve cells or neurons. There is one visual distinction of nervous tissue, having these neurons visually located there. The other is the special property of the nervous tissue is to create electrical signals and pass them on along this tissue. Super important for communication point of view. We mentioned the voluntary nature of skeletal muscles. Well, we send skeletal muscles signals to contract or relax through our nervous system. So functionally and visually, we're looking at a very distinct tissue type. So there's a single tissue, nervous tissue, in the nervous tissue category itself. So the two cell types that make up the nervous tissue are really well illustrated on this image. And I'm going to scroll this up for the first few seconds. So neurons and neuroglia are typical features. And in the function, we are going to separate neurons and neuroglia. So let me see if I can do another Zoom session here. When you look in nervous tissue, one type of cell that kind of grabs one's attention, large, dark purple, or you might say dark pink. Here's the outline of the cell. So this large cell is one of the neurons. There's another one down here in this corner. Neurons have extensions. They have axons and dendrites. Here, looks like this neuron has a long tail. This is most likely the axon of the neuron. And here you see shorter extensions. These are probably the dendrites. Neurons are functioning by creating or conducting nerve impulses. So this is a very unique function. Nothing else in our body can do this. So neurons are uniquely able to create nerve impulses, nerve signals, and move them along long distances. The axons of some neurons in the body are measuring several feet in length. Here too, we are seeing only a small portion of this very, very big neuron, because a lot of the axon is cut away. So these cells are uniquely able to create and conduct nerve impulses. How they do that, we'll analyze that in great detail in chapter 12 in lecture. For the functioning of our nervous tissue, though, we need the other cells. Well, these cells look like very dark purple or dark blue specks. You see a whole host of them here. Every tiny dark spot here represents a cell of the neuroglia. Neuroglia cells cannot fire a nerve impulse. They cannot create nerve signals. But the neuroglia cells fulfill multiple functions to support the neurons. So neurons receive a nutrient source. These neuroglia cells clean up any kind of debris that might appear in between neurons. In chapter 12, and we'll be detailing all the neuroglia subtypes. For us here, suffice it to say that these tiny cells are corresponding to neuroglia, and their function is to support neurons. So this is the night and day nature in terms of shapes of the neurons and neuroglia, and also night and day difference in function. Nerve impulses are tremendously important for our homeostasis. Our nervous system monitors, controls, adjusts many aspects of the working of the body in many organ systems. So it's impossible to overstate the importance of nervous tissue, the neurons, and the neuroglia. Any questions about these last two tissue categories and these four tissues that belong to these two categories? I'm going to check if we have any chats coming in. I don't see any new chats. So this is the best time to take one of our breaks. So this is 116 going on to 117. So let's restart the lab at 127, and then we'll go into the integumentary system. The same lab report applies. It starts at the bottom of the first page, continues on the next page, and so our first organ system will be the topic when we restart in 10 minutes. Here. So our integumentary system is at the border of the environment and the rest of the body. So there is a barrier function. We want to keep the quote unquote good things, useful things for our body inside, and we want to prevent the potentially harmful chemicals, temperature to come inside the body. So we have to have a barrier function. We'll be seeing that the top layer of our skin, epidermis, is a tremendous barrier, not only for chemical, but also biological agents that could cause harm to our body. Our integumentary system expect to stay on those underlying structures, don't come apart, resist forces, even of extreme nature. You can try to grab on your skin and pull it off your arm. It's not going to budge because our skin is tremendously resistant to many types of forces. It's resisting being taken apart, being pulled away, pulled off of the structure that it covers. So we need to have a physically reinforced portion to this integumentary system to fulfill that kind of function. Our integumentary system is expected to fulfill a temperature regulating function, and this will come from the eccrine sweat glands. These are epithelial structures, if you remember, going back to the tissue lecture. This is something that's uniquely needed for the human body. You know about other critters that have no sweat glands. Think about dogs. They control their temperature in a different way. They have big tongues, they hang out of their mouths, and their body cools down in part of that cooling action coming from evaporation of their saliva. We don't tend to do that. we have our entire skin surface area to evaporate sweat. So thermoregulation is another expectation of our integumentary system. And then we have yet another function that we take for granted. And this is a sensory function. As you're sitting there next to your computer or laptop, you are detecting the temperature of the environment around you. How is that possible? Well, we have temperature sensors in our integumentary system. You are detecting that you are sitting on a chair. We have pressure detectors, pressure receptors in our integumentary system, including the skin of our butts. So we detect that. If you type something on a screen or on a keyboard, if you pick up a pencil, you are relying on your touch receptor to tell you that you touch something, it's time to push down. So we have touch receptors. These are all elements of our nervous system. We'll come back to these in chapter 12 in a general look, just how well our nervous system depends on gathering information about the environment, and sensory portions of our nervous system, such as these receptors, are integral part of the entire system of monitoring our environment and responding to this change in the environment if we need to do that. So many functions are coming out of this relatively simple organ system. So let's take a look at how we can connect this to tissues and models and move on all the way to the appendages. We're going to start out with the two basic parts of our skin, which is epidermis and dermis. Let's see if I can do this somewhere here. So on the second PowerPoint slide, here's a very simple structural division of our skin. So our integumentary system will need appendages in addition to skin, but skin provides the basic scaffold structures to our entire integumentary system. So it's a good place to start. Epidermis is the surface layer. So this is meeting the airspace by tissue type. This is epithelial tissue, stratified squamous epithelium. As we'll see in a few minutes, it has several individual layers within the epidermis. The dermis is much thicker. It's deeper. We have two regions here. We have a papillary region with areolar connective tissue. And the reticular region, I put the name of this connective tissue here. This will show up in lecture. We didn't see this in lab. Thick collagen fibers provide resistance to force in this tissue. If I go to this screen share right here, our lab show, this is our integumentary system model. And here you see the strata of the epidermis. And here you can see bracketed the epidermis. So this is the air interface on top of our skin as you look down on it. We'll talk about these layers in a few minutes. The other major layer of our skin, dermis, is this pink plastic every place you look. Here is the papillary region bracketed, and the reticular region makes up the rest. If I scroll down here, you see that there is another thing named here hypodermis. This is rich in adipose tissue, blood vessels, hypodermis does not belong to our skin. It is just glued to it. And so you'll see this in lecture material as well. Hypodermis is labeled here. It's not part of our skin. It's not a third layer of our skin, but it is directly deep to the reticular region of the dermis. You see a whole bunch of other structures here, including hair follicles, these mustard yellow structures, these grayish coiled garden hose looking structures. Those are all appendages, and we'll talk about them the last part of the lab. So when this skin model will come your way on a practical, it's important to be able to name these strata, the corneum, lucidum, basale. It's important to be locate the dermal papilla and to identify the reticular region. The top layer of our skin is stratum corneum. I'm sorry, the top layer of our epidermis is stratum corneum. The deepest layer of our epidermis is this orangish layer, meandering layer, the stratum basale. The model also points out the stratum lucidum layer. This comes about through a special development of cells in this layer. It's the story of food deprivation, oxygen deprivation, and how the cells respond to this. So to explain these layers and follow that up with a microscope slide, let's go to this last PowerPoint slide here. And if you have any questions, please don't hesitate to ask. And let's see, I'm going to have to check. I don't see any more chats.
Can you describe, I'm sorry, could you describe not describe, but name the 3 layers of the stratum that you'll be required to know?
Oh, I'll mention these on this screen share too, and we can go back to that too. So on a model, what you saw is some strata labeled. So the epidermis is this region of our skin, superficial to this black line, which could be the basement membrane. Deep to that is the dermis. We're not going to talk about it here, but let's take a look at how these layers come about, these strata. We have a exceptionally well-living layer in our epidermis, the stratum basale. This is one of the layers that you're expected to recognize on the model. This is that orange meandering line on the model. The stratum basale layer cells have everything going for them. Remember from the from the epithelial portion of the lecture, epithelial tissue such as this stratified squamous epithelium have no blood vessels. So all nutrients and oxygen need to diffuse from the dermis. And stratum basale cells are very close to this flow of nutrients and oxygen. They live really well, and they have the energy, the nutrient source to divide. So stratum basale cells multiply 24/7. We'll come back to this in a lecture. I'll match it with some other parts of our integumentary system. So these are the cells some people call this the stem cell layer of our epidermis, constant cell division, huge flow of nutrients and oxygen supply all the needs of the stratum basale cells. When the stratum basale cells divide, the daughter cells are pushed superficially. So after two cell divisions, this green cell layer forms on top of the stratum basale cells. Remember, these cells keep on dividing, and these two cell layers in green are now pushed away from the source of nutrient and oxygen flow. The stratum spinosum cell layers still survive. They have enough nutrient and oxygen trickling through the stratum basale, but these cells don't have enough nutrients and oxygen to divide. So stratum spinosum cells just sit there and start making a few proteins and lipids, but they don't divide. After two more cell divisions of the stratum basale, the green cell layers will be pushed even further away from a nutrient source, and they turn into this yellowish cell layer, the stratum granulosum. In this cell layer, the cells are barely alive. They are storing away, stashing away in granules, lipids, and a protein called keratin. And when we look at the microscope slide, you'll see a fairly dark appearance of this, the cells because of a large number of granules. This is a stress program in these cells. And a few more division of the stratum basale, and the stratum granulosum cells are now pushed even further away. They will go totally dead, and they form this dead cell layer, the stratum corneum, in purple. So when you look down on your arm, you are looking at your stratum corneum layer of the epidermis. Cells are totally dead. They still hold on to each other for a good deal of time. And so it's this layer that provides the best vapor barrier, the best barrier against pathogens, whether it's viruses, bacteria, fungi. We have millions of bacteria landing on our stratum corneum, but they have no food, they have no water, so they don't divide, they just sit there totally harmlessly. So our epidermis builds and fulfills this barrier function thanks to the constant division of stratum basale. On the model, the stratum corneum is visible on the top of the model. And you know what? I'm just going to go back to that stuff, talking about that right here, because I'm going to come to the lab shell anyway from microscopic view. So here's the stratum basale, stem cell layer in our epidermis. The stratum corneum is always this top layer. There is in some parts of our body, we have a skin called a thick skin, and it has this reinforcing layer, stratum lucidum. This is a layer of totally dead cells. They are totally bright, and this is why they're called lucidum under the microscope. And so those are the three strata that are easily visible on the model. So lucidum, corneum, and the stratum basale. We're not going to worry about the spinosum and granulosum cell layers on the model. When we look at a microscope slide of the epidermis, and again, I'm going to project this from the lab shell. This is titled Epidermal Layers. And here you can identify all the cell layers, all the strata. I'm going to zoom in a little bit here. So we see the dermal papillae and the papillary region of the dermis down here. It's not labeled, but this is what these arches are. And you can see the single cell layer, the stratum basale, going all in this meandering fashion, just like on the model. Superficial to the stratum basale is this large number of stratum spinosum cells. Again, they're still alive, but they cannot divide, so they are being pushed further up away from the nutrient source. Here's that stratum granulosum layer, much darker. If we would have a high power microscope, we could see the granules making these cells dark. This particular human skin was obtained from a part of the body where reinforcement is needed, and this is usually the palm of our hands, bottom of our feet. The stratum lucidum layer is a much brighter layer, just superficial to the stratum granulosum. Again, the cell layers came about because we are gradually depriving cells of nutrient and oxygen. So the cells slow down first, and then they start dying and creating granules. And by the time of stratum lucidum, cells are totally dead. Further division of the stratum basale will create a thick top layer of stratum corneum, as you see here. Some of the proteins that are accumulate inside the cells are called keratin. And this process of moving from stratum basale all the way to the stratum corneum, this is called keratinization. And the cells are called keratinocytes here in our epidermis, whichever layer you look into. So this is the formation of this keratinized barrier layer. Again, stratum corneum is the most important part of this barrier layer.
Would the cell type be keratinocytes if there was an arrow pointing to that area?
So the individual cells are called keratinocytes. It's more important to recognize these layers. Any other question about these layers of the epidermis? So we don't have a microscope slide about the layers of the dermis. Most every place you see the papillary region with a little bit of the reticular region. Again, this model is the best way to point out the reticular region and the papillary region. Inside the dermis is where we see a large number of these appendages. So if you look at the lab report, the dermis has a question mark or a question here, or request list layers superficial to deep. So within a dermis, the superficial layer is the papillary region, a deep layer is the dermis. In the first lab, we mentioned these directional terms, superficial, deep. This is why those directional terms come so handy for the skin, because this is whether relationship between the strata or the relationship between the regions of dermis. This is the best way to express that position.
I'm sorry, but could you just list them really quickly from deepest to superficial?
So in terms of dermis, the deepest is the reticular region, and the superficial is the papillary region.
So that you don't want the answer to be basale, spinosum, granulosum, corneum.
That is for the epidermis. So for the epidermis, the deepest layer would be the stratum basale. And on the microscope slide, you will see the stratum spinosum, stratum granulosum, stratum lucidum, and stratum corneum. It's also in that layer, in that order on the PowerPoint slides that I drew. So the most superficial layer of anything is the stratum corneum of the epidermis, right? Superficial means the closer to the outside of the body, and stratum corneum is the most superficial layer of the entire skin. So within the dermis, we have a large number of these appendages. So let's take a look at the structures in the order that they appear in the lab report. So we have here hair-related structures. So on the screen share, I'm going to scroll down here because we have, I think I'm catching, showing everything here. Let's see if I can do this. Maybe this is the best. So hair-related structures. We have the hair follicle, and in lecture we describe how the hair, that set of cells is growing out of this. So we have the hair follicle. You see this in a cross section right here, and the bulb region is this wider base. As the hair is constantly growing through this cylinder-shaped hair follicle, the portion of the hair that's still inside the skin is called hair root. This pictured here as well. Once the hair is protruding out of our skin, this is called a hair shaft. The hair itself, the hair follicle, is an attractive place for many other structures. If you want to move the hair follicle, we have this reddish arching structure. This is smooth muscle. This is called the erector pili muscle. And this is serving the movement of the hair follicle. Movement of the hair follicle when the hair stands up is causing goosebumps. So that's the only time when you can actually visually see the effect of the workings of the erector pili muscle. Right next to the hair root, we see these gray structures. These bag-looking structures are the sebaceous glands. So they have a secretion which is called oil or sebum, and that secretion is poured right onto the hair root. It kind of makes sense that in the skin areas where we don't have hair follicles, we don't have sebaceous glands either. The two go together very organically. The sebum, or the oil secreted by the sebaceous glands, moisturizes the hair. And also, as it comes onto the surface of the skin, it moisturizes the stratum corneum as well. A little bit over here, you see yet another structure having an opening onto the hair root. This is called the apocrine sweat gland. The apocrine sweat gland creates a viscous, thick secretion. Again, it's poured onto the hair root. And it also has some moisturizing function. The apocrine sweat is the secretion here from the apocrine sweat gland. And then we have a different sweat gland, the eccrine sweat gland. Sweat glands look like a coiled set of garden hoses. A big difference between the apocrine and the eccrine sweat gland is that the duct of the eccrine sweat gland goes all the way to the top of stratum corneum. You can follow this here. Here's another sweat gland cut in half, but you can see that the duct is going all the way, kind of removed here to the top of stratum corneum. Here is yet another sweat gland. If you follow the duct, it goes all the way to the top of the stratum corneum. The eccrine sweat gland secretes the regular sweat or eccrine sweat, and this eccrine sweat gland and the sweat has that thermoregulatory function. This is what that helps to cool our bodies down, secretion of eccrine sweat from the eccrine sweat gland. When we get to all the way to chapter 15, we'll see the reason for what you see here. Very interestingly, bundles of axons, elements of our nervous system, are bringing nerve impulses to these glands and turning them on or off. So when we are overheating, it's a reaction from our brain is sending nerve impulse to the eccrine sweat glands and turning them on. It's real interesting how glands are under instructions of the nervous system. So again, functionally, the eccrine sweat glands are the most important sweat glands. They give us this ability to regulate body temperature. Any questions about these structures, these appendages, the hair, erector pili, hair root or hair root and hair shaft, and the two sweat glands? So the only function that we haven't addressed is this sensory function, the ability to detect touch, pressure, temperature. And so these very bright yellow structures are element of our nervous system. So this is nervous tissue. And we're going to mention only two. Your lab report has first the Meissner corpuscles. Meissner corpuscles are located in dermal papillae. You might want to take a note of this. Dermal papillae represent these finger-like invaginations. This is the papillary region of our dermis. And in many of those crevices, we find a Meissner corpuscle hidden. The Meissner corpuscle detects touch. Sometimes it is called the receptor of touch. And the other element of our nervous system that we'll mention here will be these large yellow ovals deeper in the reticular region of the dermis. There are quite a few of them. Only one is labeled Pacini or Pacinian corpuscle. And this detects pressure and vibration. So a deeper force on the skin will trigger nerve impulses from these sensory structures. And so our brain will be informed that pressure is exerted on our skin or vibration is stimulating our skin. And again, there are several Pacini corpuscles here. Any questions about the appearance of these structures, their functions on the skin model? Many of these structures have a microscope slide equivalent. There's one additional set of structures that we are going to mention. That is the melanocytes, but let's review the microscope slides of the structures that we have looked at. So I have here, again, I'm projecting all this from the lab shell. Here's hair and sebaceous gland structures. So on this microscope slide, this very thin pink purple line is the epidermis. You can see how incredibly thick the dermis is and just how deep hair follicles go into the dermis. So here's a hair follicle, now a real one, not a plastic one. Here's the hair root, and you can see a little bit of the hair shaft. The hair turned yellow under these staining conditions. And you also see this bag-like structure, the sebaceous gland, and pouring its secretion, the sebum, right onto the hair root. Model makers represented this really well. If I move this to the left, you see yet another hair follicle, yet another sebaceous gland. The hair is missing from this particular hair follicle. How do we know it's a sebaceous gland? You'll see a sweat gland appearing under the microscope. Sweat glands look like a coiled garden hose cut in half. You see a bunch of circles next to each other, totally different look. So this is the appearance of hair, its related structures, and sebaceous gland. So let's take a look at that sweat gland. This is what I meant by a bunch of circles next to each other. We are deep in the dermis, in the reticular region, and sweat glands appear like a series of ovals or circles. We don't know if it's an acrine or apocrine sweat gland. They all look very similar. At this part of the dermis, we would have to find out where their duct opens, and we just don't see that. So I just want to show this in contrast to the sebaceous gland. This is the appearance of a sweat gland that is cut for these microscope slides.
When asked to name the image on the slide, do you want sweat gland or do you want eccrine or apocrine?
Oh, sweat gland is good enough. Again, we cannot determine. So I'm not going to ask you to name specifically something that we cannot see. So this can stay a sweat gland. So let's take a look at those sensory structures, and this will be probably the least visible. Let me see. Let's take a look at the Meissner corpuscle. So here you see the entire epidermis, very similar to that epidermal layer slides, the stratum basale cells. And here in this part of this thick skin, again, you see the stratum lucidum here. This has to be a reinforced layer of the skin. Stratum granulosum shows up real nicely, stratum spinosum. But into this epidermis, we have these finger-like protrusions, the dermal papillae. Meissner corpuscles don't like to stain into any kind of color, so they just remain this barrel-shaped set of squiggly lines. So these are the Meissner corpuscles as they show up under the microscope. Again, the location is dermal papilla or papillary region of the dermis. And if you want to take a look at a Pacini corpuscle, this is also faint because the dermis doesn't stain real well. Pacini corpuscles as elements of a nervous system also don't stain really well. So what do we see here? Again, deep inside the dermis, there are these two large round structures. We have two Pacini corpuscles side by side. You can see the layers within the Pacini corpuscle. So this is what the Pacini corpuscles look like under the light microscope. And again, I'm projecting from the lab shell, and this the labeling is there. Pacini corpuscles and Meissner corpuscles are probably the most challenging aspect of all the microscope slides that we have looked at in this entire first part of AMP1. So something that we didn't see on the skin model is showing up only here on this slide. And let me see if I can find this. So this is melanin-rich skin. We'll talk about this in lecture. Melanin is the natural substance that gives color to our integumentary system. This color is naturally brown. Melanin is an organic chemical made by melanocytes. So melanocytes don't show up. They will be residing right here in between the stratum spinosum and stratum basale cell layers. You can see the dermis labeled here. This is the region of our skin here, the dermis. What happens to melanin? This dark substance is endocytosed by stratum basale cells. So the stratum basale cells gain this very rich brown color. Some of the stratum spinosum cells endocytose it too. And so on this slide, the stratum basale becomes extremely visible because all you have to do is follow the brown staining. And you can see the single layer of cells. This is, again, the stem cell layer of our skin filled on melanin particles. Again, the cells that make melanin is called melanocyte. They are not visible. They just don't stain real well. Melanocytes don't hold on to melanin. They exocytose it into the space here in between the cells, and neighboring cells take it up and load up on melanin. We'll talk about this a little more in lecture. There's a little better view of that, but think about UV light coming in from the top of the skin here. You can see stratum corneum partially broken up. And UV light would cause damage to the DNA inside the stratum basale cells. They are dividing 24/7, so damage to the DNA could be very detrimental. So melanin particles absorb some of the UV light, and so it's like a brown umbrella. It keeps UV light from hitting the DNA of these stratum basale cells. So this umbrella function is not without leaks. The most frequent cancer in the human body is the cancer of the stratum basale, in common clinical terms, basal cell carcinoma. That refers to the originating cells of the stratum basale. If you have older relatives, they may have had basal cell carcinoma just based on sun exposure. The treatment is very easy. You can cut out the part of skin that develop basal cell carcinoma. This cancer normally does not metastasize, doesn't spread. So cutting it out is the best way to deal with it. Again, if you have relatives, friends, maybe coworkers, mostly elderly, went to the dermatologist and came back with a Band-Aid on their face, basal cell carcinoma was removed, and there's nothing to it. Skin will regenerate. These stratum basale cells will feel the wound of the epidermis, and so skin will heal up. And that is the last aspect of this lab. Again, this slide too is in your lab shell. When you look in the lab shell, you will find all the epithelial and connective tissue slides there as well, with all the functions, specialized cells, location in the body, tissue category. So lab shell is a really good resource to get the visuals and put it together with the rest of the information on these tissues. And I hope your lab report will be really helpful to relate functional information with the visuals, whether on the model or on the microscope slides. Any questions about any of the models that we looked at, any of the structures that we named, any of the microscope slides that we looked at today or looked at before? Any questions about anything that we covered in this first 7 exercises for the lab? I'm not hearing anything, so let me check this chat. And I'm not seeing any other chats. So if there are no questions, we're going to close up the lab. Please remember that, again, there's no new lab home assignments. There's no new chapter quizzes due before your practical. Please focus on this. And I hope everybody will do well. Again, tissues will take the lion's share of this practical. So be ready to look at lots of microscope slides. Also from the integumentary system, we had microscope slides representing cell division. So there will be a lot of microscope-based images that will come your way this practical. So what I'm going to do, if there are no questions, I'm going to stop this recording. I'm not sure how to attach it to the class shell, but I'll try to add it to the same area where the lab report would be and where the PowerPoints would go for this particular lab. So I'm seeing a question here. Is the practical on Monday or Wednesday? Well, let me check my schedule. I'm seeing here practical June 22nd, which is a Monday, I believe. And so when you look at your schedule portion in your class shell, this should show up. The note here is we start at 9 a.m. and lecture will follow. So we will have the tissues and integumentary system lecture after that. And again, I will send a reminder email, but please join the meeting on Monday for the practical through your phone.
All right, what about the chapter review? I said,
Say this again?
The chapter review we sent, the one you asked us to email it.
I'm sorry, I'm not hearing you well.
I said the chapter review you asked us to email you. So I don't know how you're going to put it together because it's just so funny, you know, taking the picture and it's
yes. So
some of them could not attach a lot. So
some of you have yes. So some of you have sent me the extra credit information for the chapter reviews. So you have till midnight today. Yes, please take an image of those. Please take an image. When I try these different ways to send attached material, the easiest way is to send it to my email address and attach all those photos. If it doesn't go through in one go, send me 2, 3 emails. That's perfectly fine. Did I answer your question?
Yeah. Yes.
Okay. And so we were talking about the extra credit for lecture exam 1. I see another question. How many points each extra credit assignment worth? So for each lecture exam, you are earning 6 extra points. And roughly it means that 1 chapter's worth of review sheets correspond to 2 extra credit points. And yes, they are due midnight tonight, so I'm not going to look for them until tomorrow morning. Another question here, will we make up the lab quiz we did not take on Monday? No, I'm going to give everybody an excused mark for that lab quiz, so don't worry about that. No makeup of lab quiz. The only thing you'll see coming your way as a graded activity on Monday will be the practical at 9 o'clock. All right, I don't see any chats. I don't hear any other questions. So let's close the lab. If a question pops in your mind 20 minutes from now, please send me a message through the class shell or send me an email, and I'll be happy to answer or help you out. Again, if you want to request a Zoom session going over the material, I'll be happy to do this. So please prepare for this practical major chunk of points for your grade coming from this. So if there are no other questions, thank you for sticking it out through this long day exam and a long lab, and have a great weekend, and I'll see you all on Monday.
Yes. Bye.